I want to give you a little bit of background about me and the Marine program before we launch into Sea Stars. So Marine stands for the Multi-Agency Rocky Intertidal Network, and we are a group of a bunch of different organizations along the whole West Coast that are doing long-term rocky intertidal monitoring using the same methods. And shown here are some of the species that we target with our monitoring protocols. And the ochre star, which is down there on the bottom, is one of the many species that we target. And so when the sea star wasting disease event happened several years ago, we were in a good place to be documenting the impacts of that disease on intertidal sea stars and on the ochre star in particular. I'm not going to give a lot of information about what we do. There's a lot of information on our website, which I'll show people in a few minutes, but this is the span of our sites currently, and we have two major sets of protocols that we use. One is what we call our long-term surveys, and those we go back to sites either once or twice a year and monitor the same plots over time. And then the other protocol that we do is called our biodiversity surveys. And those are for capturing more of the species throughout the site, and they're done on a much less frequent basis. So that's a very quick and dirty overview. For sea stars in particular, we use permanent plots that we count and size sea stars in. The ochre star, Pisaster ocraceus, is the one that's most abundant along our coast, so it's the one that we primarily focus on. And we measure the radius of the star. We call it the radius from the center of the star to the tip of the longest or straightest arm. And that gives us data like this. This is just an example site. This is showing the sizes across the bottom, so going from smaller to bigger, and then this is just time. Oldest is at the top, the newest season is at the bottom, and these bubbles represent the numbers of sea stars that are in a given size class. So this particular site had a lot of stars. This biggest bubble represents 90 stars, and so this is sort of typical of what a site would look like prior to the disease, where we've got a large number of stars that are kind of in the 100 to 150 size. And then in 2013, you can see the stars kind of disappeared from this site. And I picked this as an example because it shows this recruitment event. You can see over here in the lower left corner, there's a bunch of very small stars that have come in, and most of the big stars are gone. And as I mentioned, that's due to sea star wasting disease, which I think probably most of you are familiar with. I'll just go through really quickly what we know and don't know about it. Mostly we don't know much about it. But it's a disease that's really a general description for a set of symptoms that have been seen in many species of stars, not just ochre stars, and historically other echinoderms. So this is something that has been kind of cyclically around in Southern California, the term wasting disease. We don't know if what we saw coastwide is the same thing that we've seen on a more cyclical basis in Southern California, but those previous events were much smaller in scale than this big event that we're probably all familiar with. But in all cases, there's never been a definitive pathogen that's been identified. And so we really don't know what's causing this disease, which is kind of frustrating because then we can't test to see whether stars actually have the disease or not. But we know for sure that it's present at a site when stars start to look like this one on the left, where they've got lots of tissue decay, and there's lots of individuals that are dying. So there's lots of ongoing work still looking into potential causes for this this disease. Some of them are looking at the microbiome, so viruses and bacteria. Others are looking at possible associated factors like temperature or ocean acidity. We know that there's over 20 species of sea stars that have been affected by the disease. And it's kind of persisted. It kind of comes in waves where we see it and then it kind of disappears, but it seems like it's always been present in most areas really since 2013. And on a very broad scale, we haven't seen a strong temperature link, but there does seem to be a correlation at a more localized level. So this is all of our sites, and I don't expect you to read all the site names, but generally, hope you, on my screen, this is covered up here. Hopefully other people can see this, but this is all of our sites going from north to south. And I just want to show you this big scale picture that we've seen with ochre stars along the entire coast. And then I'll talk about Oregon a little bit more in more detail. But these range from Alaska all the way down to Southern California. And the regions are listed here. These are individual site names. And each of these boxes represents the population size at a site relative to the mean prior to the disease. So this line here represents when we first started seeing sea star wasting disease along the coast. And so you can see that in most regions, the populations are represented by these paler, kind of whitish or blue or purple colors. And that's, kind of at the mean level or even sometimes a little bit above the mean. And then after this line, so this is after 2013, most of the sites are red. The populations of the sites are red. And what that means is we've had decline, and in some cases, really serious decline. And I've drawn this other line in here to represent a break at Point Conception in California, which is a really important biogeographic break. And this is kind of a spot where we see big differences in the pattern of both decline. So maybe a little hard to tell, but the level of decline is significantly less at all of these more northern regions, so from Central California north, and then generally is more severe in these Southern California regions. And I said I'd talk about Oregon in more detail. So this is what Oregon sites look like. So this is again from north to south, and you can see prior to 2013, this is sort of the normal state where populations fluctuate, but are generally within that range of normal. And then we saw decline in Oregon. So there was definitely a decline, but it wasn't as strong as a lot of the other regions. And we're starting to see some of these sites come back to pre-disease levels in terms of sea star numbers. So this is Bob Creek, just south of Newport. And this site in particular has had a lot of recruitment and a lot of recovery. So in general, I'd say the Oregon coast is looking pretty good for sea stars. This is the other piece of the picture. So this is the recruitment patterns that we're seeing coast-wide. So again, from Alaska to Southern California. And this is even more striking than that decline pattern. So you can see here's that line that separates California kind of right in the middle. So this is all Southern California south and then Central California north. And the darker the colors here, the more recruitment we've seen. So the more juveniles we're counting at sites. And I'll zoom in on Oregon so you can see what's going on there. And you can see this scale ranges from 0 to over 600 stars. These are juveniles, so individuals that are about slightly larger than, I don't know, maybe the size of 1.5 or 2/4 or smaller. And so we get large numbers at several sites, but it's interesting that that's not the case across the board. So even within a region like Oregon, where things are looking pretty good, there's a lot of variation in recovery. And then this last thing, this is my signal to myself to switch to the website. This is a site that's monitored by your group, actually. It didn't start until 2015, monitoring at YAHATS. So you can see that numbers started off pretty low. This green line is showing Ochre Star numbers. But things have been recovering nicely over time. So this last survey was in August of 2019. And so things are looking pretty good at that site. And I know there's a group associated with Oregon Coast Watch that's been monitoring this for several years now. Is there anything else I wanted to say about that? And I guess before I move to the website and talk about some of the resources we have available to you, maybe I should see if there's any questions about sea star wasting or kind of those coastal patterns in general. And Jesse, I'm not actually sure, is that something, do we want to wait till the end for questions? Is that easiest?
Oh, we can definitely take questions. I see that Fawn is here and has a question. So I opened up the Q&A, and yes, if anyone wants to ask a question, could it be, let's see, can you see the questions too?
Let's see.
Melissa?
I have a question and answer thing, but I don't see any questions right now.
So it says that to all panelists and attendees, so maybe you might have to open it up, but I'll read it to you. Could it be that Bob Creek, Fawn asks, could it be that Bob Creek is more remote? And then Jill asks, were the other species returning like the disaster?
Yeah, so I'll answer Fawn's question first. I would say, so if I back up a little bit, let's see, can I do that? There we go. So Bob Creek, I think it's just a great spot for recruitment. Fogarty Creek, I would say, is our most remote site. I don't know, even Burnt Hill, I mean, that's right on the border of Oregon and California. It's a really challenging site to get to. So my guess is it doesn't get a lot of visitation. Bob Creek is, I would say Bob Creek, Ecola, and Cape Arago are probably all about the same in terms of visitors, although every time we're at Bob Creek, we see people who are out there fishing or tide pooling. So yeah, I don't know that I would say that it's due to remoteness. I think it's just a good site for recruitment. And I know Bruce Mengi has done a lot of work looking at recruitment patterns on the Oregon coast, so that would be a good resource to look for his papers to see what the general trends are for recruitment along the Oregon coast. And then I'm sorry, I'm forgetting the second question.
Oh, Jill asked, were the other species returning like the ochre?
Oh yeah, so I mentioned at the beginning that we focus primarily on ochre stars because they are the most abundant, or tend to be the most abundant, in the type of habitat that we target, which is the rocky intertidal, so rocky intertidal benches. The other species that can be abundant in the intertidal on the Oregon coast is the six-armed star, Lept Asterius. And we have data for those as well, but they don't go back as far. That's something that we added more recently. But we have definitely seen declines in those. And, you know, offhand, I can't say what populations overall for the 6-star star look like. All species have been affected by this disease to some extent. Kind of anecdotally, it seems like leather stars are maybe the least impacted, but we do see individuals even of leather stars with signs of disease. So yeah, I would say, this is the species that we have the best data for. Most other species tend to be rare in the intertidal. At the end, I was going to talk about the sunflower star, Pycnopodia, a little bit, because there's been kind of a special focus on that particular species, because it has experienced such a significant decline across pretty much its entire range. But we just don't see a lot of those in the intertidal. So it's not something that we have a lot of data for from this particular project.
Okay, thank you. We have a couple of more questions. Is it a good time?
Yeah, and now I see, is it known, this is from John French. So that's the only question I see for some reason.
Okay, well, Stacy's question was first, and then I gave her a little private answer that we would get to it next. So why don't we take hers and then we get to John. So Stacy asked, for a while, the leading idea behind sea-star wasting disease was adensovirus. Was that disproven?
So that was work by Ian Hewson, and a bunch of us were actually on that paper just because it was kind of the first paper that discussed the patterns. Some of our data were included in there, as well as the cause. And it's still very much up in the air. Ian Hewson had a subsequent paper where he showed that it was unlikely that it was the cause for any species other than Pycnopodia, the sunflower star. And, in my mind, it seems unlikely that adensovirus would cause the disease in one species, but not the others, because this was, it affected so many species so broadly. I think that, my personal opinion, and I'm not a disease biologist, but is that it has to be linked somehow. So there's got to be the same kind of underlying cause, and there might be locally different drivers that kind of ramp up the disease or make it less virulent. But it's still very much unknown.
Okay, so you can see John's question. Is it known what makes a good site for recruitment versus a site that's not doing so well?
Yeah, so a lot of it is related to upwelling patterns. So when there's strong upwelling, the water on shore is actually pushed offshore. So that's, part of the picture. There's lots of factors that go into whether or not larvae can recruit to a site. So it's, it's kind of species dependent too. Some of them are passive in, meaning that they can't actually control too much where they go. They're more controlled by currents and, other oceanographic patterns. Others of them actually can move and spend more time feeding. Some of them feed, some of them don't feed. So I would refer you, especially along the Oregon coast, I would refer you to Bruce Mangy's work. He's really the expert for recruitment patterns in Oregon.
Okay, thank you. All right, looks like we don't have any more questions at the moment.
Okay, so I'm gonna switch now to, let's see. To switch to the website so that I can share some of the resources we have. So this is our marine website. This is just the general website page. In terms of C-stars,
Oh,
the thing.
Melissa, we're just seeing your slideshow still.
Oh, you can't see the,
So
well.
maybe you just need to switch your share screen to your.
Oh, it says my screen sharing is paused. I
Oh,
wonder why.
I wonder if you accidentally paused it. Let me see.
Oh, resume share. Here we go.
Okay.
Okay, can you see the website now?
No,
No.
we're just seeing your slideshow. We're seeing the last slide that we were on, the recruitment patterns.
Well, sorry, I thought that it was just my screen.
Maybe just close your slideshow.
Okay, I was gonna go back to it, but I can open it up again, I think.
We have, oh, there we are. Hey, everybody.
All right, let's see.
We have some of the attendees offering suggestions as well.
I'm sure, yeah.
A lot of folks have been in these Zoom meetings, so.
Let's see. Well, let's see, share screen.
And if you can't, you should be able to do it. And if you can't do it, I can also probably get to it as well.
Well, I've got share screen, and then how about that?
There we go. It worked. Okay.
All right.
Super.
So we'll see. If I can't get back to the PowerPoint, that's okay. So this is our general website. There's a lot of information here about the monitoring program in general, but in terms of C-stars, the best place to go, and I had this on the PowerPoint, is if you just type in cstarwasting.org, that'll take you to this page as well. And there's lots of resources here about C-stars, the disease. We have identification guides that you can download, both for species, and we've got good juvenile identification guides, because it can often be tricky to tell the tiny stars, tell what species they are. And the thing that I really wanted to share with this group is this feature we have where you can submit observations. So you can go anywhere along the coast and then submit observations about C-stars that you see. And unlike the population monitoring, where it's very specific, you have to go and monitor exactly the same spot every year. You have to be committed to go every year, and and make sure that you can sample in a way that's consistent with the rest of the monitoring on the coast. This, I think, is much more fun and relaxed. So you can submit observations about any stars that you see. And we have recently switched this to calling it just our C-star map. So what happens with these observations is they all go onto this C-star map. And this is a really great resource, both for the public and for researchers. We get a lot of requests for this information. And if we go, I'll just choose a site on the Oregon coast. So if we go here to Cape Perpetua, you can see the last time anybody went there or sent us an observation from it was in 2014. And so that's why, in talking to Jesse, I was thinking, wow, it'd be really great to share this with your group and get more recent observations along the Oregon coast. And again, this is something you can do anywhere. There's no time restrictions. You can submit observations once a week, or you can submit them once a year. You don't have to go to the same spots, or you can go to the same spot every single time. But having these broader scale, for our long-term monitoring, we only have 5 sites that we go to every year on the Oregon coast. And so you can see there's many more observations along the coast here from these from the observations that have been submitted. And it gives us, especially during the beginning when there was a lot of interest in making these submissions, we got a lot more frequent observations, which was really great for people who wanted to try and do modeling exercises with the disease, because then they could look at some of these physical features and how when people were seeing stars start to show up as being sick, what might be different about the environmental patterns. So I will show you how you can submit observations. Let's see. Oops. Go back. Sorry.
It's great.
Okay, here we go. So if we go to submit observations, if you click on this disease observation log, and again, we were focused on disease early on, but really what we're interested in now is knowing whether you see healthy or sick stars of any or all species that you see. So it's really not a disease thing anymore. It's more of a whole coast sea star map. And we ask people to enter kind of particular information, just because that that helps us to know exactly where you saw the sea stars, and we like it if people are able to ID species to or sea stars to species, if possible. If you can't ID a species, or if you're not sure if it's sick or healthy, you can definitely send in photos. But again, we've got all those identification guides that you can use as resources. And I know there's lots of other good sources of information for learning your sea star species as well. So the information that we ask for is just some general location information. So we start off with the sample date, so when you were actually there, what the site name is. Oftentimes sites have local names, and then the region where so for most of you that would be Oregon, and then the latitude and longitude information in decimal degrees. And this is something you can get off of Google Maps. And then location type. So this doesn't have to be rocky intertidal. This has been one of the really great things about having this as a way of getting information, additional information about sea stars, because for our long-term monitoring, as I mentioned, we focus entirely on rocky intertidal benches, but sea stars live in lots of different habitats. And so we've got observations that people have submitted from rocky shores, cobble shores, sandy beaches. We have people who dive and send us their observations on a regular basis. And then we even have observations from aquariums and other captive situations. And then we ask whether or not you saw disease at the site. That's just a simple yes, no. And then for each of the species listed below, you would just enter whether or not, if you saw the disease, whether it had signs of disease, that would be a yes. Or if they all looked healthy, then you'd enter no. Or if you didn't see any ochre stars, then you would click species not observed. And you do that for each of these. These are the species that are kind of the most common, most commonly seen and most easily identified along the West Coast. We're also looking for observations of the purple urchin, because that is a species that we've actually seen disappear due to a disease in Southern California. And we don't think that that's the same thing as what's affecting the sea stars, but it's another species that's of interest. And then there's a list of other species here that you can click on as well if you see them. These are ones that tend to be less common, but this is the leather star here, Asterius. So in some places that can be common as well. So it seems like a lot the first time, but especially if you're doing this for the same site over and over again, a lot of this information you only have to fill out one time. So you can just answer this question here. Have you submitted observations from this location before? If you check yes, then you don't have to enter the latitude and longitude information. So it saves you a little bit of work. But again, this is something that our group has used to explore possible relationships between disease emergence and some of these physical factors like changes in temperature and other things. And then we also get a lot of requests for this data set from other researchers, and it gets used a lot with students. So it's been a really great data set to have, and it would be even better with more observations. So that's kind of what I have. I can show some of these other resources on the website, but maybe we should see if people have questions about collecting this observational data.
Yes, please share your questions if anyone has any about submitting observations. It's so interesting to know how many sites haven't been observed for a few years, even though I know that there is one site, maybe two sites in Yachats, at least one that is observed every year, that is surveyed every year. But then one right really close to it is not. And it's just so interesting to me. So we definitely have a lot of folks down in that area who want to get involved. Okay, Fawn asks, will the site accept earlier dates and photos? That's a great question.
Definitely, yeah, it doesn't have to be a current date. And we encourage people, in fact, we just had somebody from Alaska who had a whole backlog of observations that she just entered, and that's really great. I mean, especially those repeated observations from the same area are really nice to have.
Okay. Jill asked, do you like photos of all observed each time?
So in general, if it's your first time submitting, it's nice for us to see the photos just to make sure that things are being identified consistently, and especially if you think you're seeing signs of disease, it's good for us to see the photos and just make sure that what you're calling a sick animal is consistent with what we would call a sick animal. There are kind of differences in how people define the disease. So there are some groups who, when C-star arms start to curl up and they just look kind of, I don't know if people have seen this, where they just look kind of uncomfortable. That is something that some groups consider an early sign of disease. And I think it can be, but what we decided is that's kind of subjective, and also much harder to know for sure that a star isn't just in a weird position versus starting to get sick. And so we really like to, before we call something sick, we like to see clear signs of lesion or tissue decay. So yeah, I would say especially the first couple times you're submitting observations, it's great to get those photos just so we can make sure that we're all being consistent. But then after that, I would say that in general, you don't need to send photos unless you have a question about the species or whether or not it's sick.
Okay. Thank you. And Karen asks, besides yes, no disease, are there actual number of stars for each species?
We don't for these because we want it to be more of a kind of general observation for a site or a log of your observation, I guess. Um, if people are wanting to do counts and sizes, then that's kind of taking it to that next level where you want to establish a set area where you're doing those counts and measurements in the same place, in the same way every time. And so that's kind of a whole different beast and requires a lot more commitment, and definitely something that we could talk about if there's additional sites where people would like to commit to that type of survey. But that is significantly more effort than this would be.
Exactly. And Karen has been involved in, I think, the Otter Rock counts. And that's why we're opening this up to others so that we can maybe even get even more observations. But yes, if we have that kind of interest, we'll talk to you about it. But
yes, yeah. No, it's great. I mean, it's great if people want, if there is interest in that, then yes, we should definitely talk about it. We do have, I would say, between our sites and Bruce Mangie's work, we have pretty good coverage in terms of, and the sites that your group does, and the Nature Conservancy, there's a number of citizen science sites that are ongoing. And so that's another option if people want to get involved with that more detailed level of data collection. I would suggest trying to get involved with a group that has ongoing surveys before you try and venture out on your own, just to make sure that it's something that you want to commit to.
Okay. Let's see. Can you see all the questions from the chat?
You know, it's funny. I mean, I've done several of these Zoom presentations and I can always see the questions, but for some reason, I have a question and answer box, but the only one I'm seeing is the one about recruitment. Okay. And I don't know why that is.
Okay. Well, we'll work on that for next time.
I'm sure it's something that I'm doing wrong.
I have a question just about that interesting line between Point Conception and South. And when I've been on the coast and doing different surveys and interpretations, a lot of people have asked if the die-offs have anything to do with warmer waters, with ocean warming. And as we go south, of course, water is getting warmer, and we're also seeing warmer temperatures every year on the Oregon coast as well. Has that been, I'm sure that's been looked into, but what's the latest on the theory that warmer ocean temperatures are causing these die-offs?
Yeah, so the temperature thing is really interesting, and there's definitely kind of two camps. There's the people who feel like it must be related to warming waters, and then there's other people who feel like there's probably two different types of sea star wasting disease. There's a warm water wasting disease, and then there's this other disease that we saw occurring along the whole coast. And the reason is that in Southern California, I mentioned we were seeing these cyclical die-offs, and those were always timed with warm water. So we'd have an El Nino event and we'd see these massive die-offs of sea stars and oftentimes other echinoderms, and then the water would cool and the disease would go away. And this was something that would happen, you know, around every 5 to 7 years or so. And if you look at long-term data from our sites in Southern California, you can actually see that populations were pretty depressed for quite some time, just due to this cycle of disease. And you know, sadly, populations were recovering in a lot of areas right before this more recent disease event happened. And in Southern California in particular, this die-off, this most recent die-off occurred when waters were cold, so it was during the winter months.
Hmm.
Um, and on the Oregon coast, Bruce Mangy was following it much more closely than we were. As I mentioned, we're only there once a year during the summer months, and he's pretty much sampling year round. And he felt like waters were still pretty cold on the Oregon coast when they first started to see the disease at those sites. So it's
That's
kind of
so interesting.
it's kind of this interesting thing where in some places it seems like maybe there is a connection, but in other places it really doesn't seem to add up. There was a recent paper published that that was a model of the disease, and I think they did find a potential link to temperature. I haven't actually read it yet. It's on my to-do list. So I don't know. I would say the jury is still out, but there definitely is not a clear link to temperature coastwide.
That's interesting. Okay, another question from John. Will we be able to identify species through your website?
Yeah, so let me pull up. I would say, there's lots of good field guides out there, and that's probably, oops, sorry, it's gonna take me a minute. Field guides are probably your best place to start, but we do have some nice resources, especially for disease. So we've got these guides that show you what 6 stars look like and the disease categories that we use. So we've got that for Physosterocritius and also Asterius, the mottled star. Oof. And then this is one that I think is really a nice guide for identifying species and also identifying juveniles, which can be really tricky. And that's something that we're really interested in having more observations on is recruitment. So this is something that I would say isn't really available anywhere else. There's certainly field guides that may have better pictures or more variety of pictures than we have here of various species. But we do have good descriptions of species, and we've got pictures of both juveniles and adults for a number of species. And this is something that, like I said, is available on our website. You can download it and and laminate it and take it out in the field with you. So yeah, I would say our website is another resource that you can use for using to identify species.
Kristen asks where the juveniles can be found. Do they live in the same places as adults? And is there a season of the year that has more juveniles? And yeah, I can answer part of that. Juveniles are found in the same places as adults, scattered around on the rock. And I've seen them also on kelp. I've seen them in a lot of different places. So do you agree they're found in the same places? But I don't know about a season of the year that has more.
Yeah, I would say yes and no. Juveniles can definitely be really cryptic, and they tend to be in crevices or like ochre stars. We often find them kind of wedged in between mussels. So the best way to find juveniles is if you have a good flashlight and you can really, look in those dark spaces, because they like to be kind of wedged in where they're protected from waves and predators and other things. So yeah, in general, you're looking, if you go to the Rocky River Tidal, you're gonna find both adults and juveniles, but adults are often out in the open more than the juveniles are. Not always. I mean, I definitely have seen juveniles out in the open as well. But you really, to have the best chance of finding juveniles, you want to have a light so you can look in those crevices. One thing, I don't know if it's worth going back to my PowerPoint presentation, but I was going to talk about Pycnopodia, the sunflower star, a little bit, because that's a species that we've seen just massive declines across its entire range. And they've pretty much disappeared from Southern California. We haven't seen any from, I'd say, Central California south. But we are starting to see some juveniles up here where, I don't know if I mentioned this, I work at UC Santa Cruz, but I actually live in Bellingham, Washington. So I'm up here in the Pacific Northwest. And here in the Puget Sound, Salish Sea region, there are some locations where we're starting to see juvenile Pycnopodia. And then also in Alaska, we've had, it seems like the areas in Southeast Alaska especially have had some pretty significant recruitment events of Pycnopodia. And we're thinking that those might, this may not be the only place they recruit to, but that eelgrass beds may be an important juvenile nursery habitat, because that's where a lot of these observations have been made, is either within eelgrass beds or in places where there are eelgrass beds nearby.
Well, that's very interesting about the eelgrass beds being important juvenile recruitments. Yeah. We'd love to see those come back.
Yeah, have you heard, I haven't seen anything on the Oregon coast about Pycnopodia, how they're doing. I mean, I know subtitle surveys in general are tricky along the Oregon coast just because it's so rough most of the time. But I know there are a few groups who, like the Oregon Coast Aquarium, for a while they were sending us observations, but I haven't heard anything recently.
Yeah, Fawn says she has not seen any in the tide pools for years. Stacy says we found one sunflower star at Yaquina Head about 2 years ago, had heavy signs of the disease, unfortunately. Jill says she has not. I have not seen them. Something that you said is just for our guests might be interesting. You mentioned the subtitle. So these sunflower stars would be found out a little bit further than perhaps the ochres. Is that correct?
Yeah, so they are definitely more of a subtitle species.
Yeah.
You can find them in the intertidal, or you used to be able to. And they weren't necessarily rare, or I guess that's not the right. It was not uncommon to find 1 or 2 sunflower stars in the intertidal on a good low tide. But like some others, I haven't seen a Pycnopodia in the intertidal for years, even up here. So yeah, they were hit really hard. And there's a group out of I don't know if people are familiar with Sarah Graham's name. She's a postdoc at OSU, and she is actually leading up a group that is working on getting Pycnopodia listed. It's ICU, I'm forgetting right now. It's an international listing that sort of alerts people to the fact that species are in trouble. So it's different than our endangered species list here, which has pretty strict requirements. But that's something that she's heading up, is a kind of a Pycnopodia recovery group.
Oh, that's so interesting. Yeah, a number of us do know her or have seen her speak, but I didn't know about that with this sunflower stars. That's super interesting.
Yeah.
Very interesting. Good to know. Good to know. And it's also good to know that like I typed a note to everybody a few minutes ago on the chat that the great thing about these observations are they can be done year round, not necessarily
Yes.
in a low tide. They can be done anytime because you know, oftentimes you can observe the ochre sea stars at a mid tide and sometimes even at a high tide.
Yeah.
But we will, as like with Coast Watch, I will get the message out to our volunteers to also be just to really keep an eye out for those sunflower stars during those minus tides. That would be a really good one to look for, especially.
That would be wonderful because there is a lot of interest in sunflower stars. And like I said, we have pretty good data for ochre stars, but a lot of these other species we don't have good information for. And there's a lot of interest in sunflower stars. The other species it's a more challenging species to find and to keep track of, but the six-armed star, the Lept Asterius, which is always pretty little, that one there's a lot of interest in by a group out of San Francisco State University, because they are what are called brooders. So most sea star species, they release their gametes into the water, and their juveniles can travel, or their larvae can travel pretty long distances before they settle and settle out of the site and grow up into adults. But Lept Asterius, they actually brood their babies. If you find them, oftentimes in the late spring, early summer, if you carefully turn them over, you'll see hundreds of little teeny tiny, like just bigger than a speck size Lept Asterius that the mother is brooding. And so they just crawl away. So this disease has been, it's been really interesting to study the patterns of decline for that species, because there are places where they seem like they've disappeared entirely, and then other places where they're doing better. And so this lab out of San Francisco State University thinks that maybe there are certain subspecies that are maybe more resistant to the disease than other subspecies. So that's another one that's more of a challenge, but it's one that there's a lot of interest in as well.
Yeah, and I actually see those not often, but a lot more often than I see the sunflower star.
Yes. Yeah.
And have some great photos of them, some of them from Otter Rock and Haystack Rock, but you can see them. And are they more subtidal as well, just like the sunflowers?
No, they're intertidal. They're really patchy. So oftentimes you'll find one and then you start looking around and there's 20 more that are kind of right near it. They're just very cryptic. They often are on the undersides of boulders. For our long-term monitoring, we don't turn over boulders. We don't move anything because we're going back to the same spot over and over and over again. And so we want to have the least disturbance possible. So that's actually, if people, I know some of you are collecting this, the quantitative data, and so you're familiar with the protocols where you don't turn over boulders. But if you're just out there, tide pooling, it's perfectly fine to turn over a boulder and put it back carefully. But that's often where you find them is on the undersides of rocks.
Okay, good to know. I love those ones. It's really exciting to find those six-armed Lept Asterius. They're beautiful. They're little silvery light purple when they're young. They're just beautiful. So thanks for that. Okay, well, we are coming to the end of our hour, and I just wanted to be able to close us out before we get shut off. I'm not quite sure if that will happen because I put in for 1 hour. So if anyone has any more questions, please ask them now, but you can always send an email to Melissa. Is that all right, Melissa? Would you like?
Yeah. All my information's on the website, and I'm happy to answer questions.
This was really great, Melissa. Thank you so much for joining us tonight. And it's nice to meet you kind of in person in this way.
Yeah, I know it's a strange world right now, but yeah, thanks again everyone for your interest in sea stars and monitoring.
Yeah, I think this is going to be a great opportunity for folks to do an observation and send in a survey whenever they want to. I think it's wonderful. And one quick question that I had was, that map you're showing, when we click on, it shows the most recent observation. Is there a way for us to see the observations up until that point, or do we have to email you to get them?
Yeah, so this is one thing, actually, I'm glad you asked that question, because one of the things that we're looking for, we're in the process of kind of doing a major overhaul on our website. And so when people are using the website, there's certain features that they would love to see incorporated. We'd love to get that feedback.
Okay.
So one of the things that, scroll down, so you can search by, if you know the site that you're looking for, you can search by site, you can search by whether or not disease, whether you want to see sick or healthy observations. You can search by species, but if you are interested in a particular site and you know where it is, you can click on that, and then you can see when the most recent observation was, who submitted the observation, and then if you go to site observation history, this will give you all the observations that have been submitted for that site.
Wonderful.
And a list of all the species. We're always happy to share this information in a more usable format if there's, if people want these data for a site or a handful of sites or a region, that's really easy for us to share. So yeah, that is how you see the history is by clicking on that link.
It's great to see some schools involved. What a great opportunity for a school to go to the beach and check out some sea stars and send in an observation log. That's just wonderful.
Yeah.
Cool.
Yeah. No, I think this is the most fun way to participate.
Yeah. Great. Well, if we have, I don't know if we have any more questions, but thank you very much. We're at the end of our hour, and it looks like it didn't kick us off at 7, so that's good to know for the next one that I do for an hour. So thanks a lot for joining us.
All right.
And thank you everybody for coming tonight. We'll see you next time. Please email Melissa or myself if you have any questions about this, and we'll see you on the beach sometime. Thanks, everybody.
All right.
Bye, Melissa.
Bye, everyone.
Thank you.